Well, there are a couple of ways in general that one can improve delivery to the brain. One is by manipulating the drug. I am not a medicinal chemist. I have no ability to do that. I know the history of that has been quite challenging. Very often, the manipulations you make to the drug to make them more brain penetrant actually remove the activity. There are some new drugs that are being developed that truly are brain penetrant. Some of them have some side effects in the brain that are a little worrisome. Some of them may not, and we'll have to see how that goes. But that's certainly an area of active investigation. There was some interest in using the drug and protein transport mechanisms to piggyback into the brain. That has not really worked out so well. Blood-brain barrier disruption, I was involved in that early in my career. It was a little bit messy, I would say, and didn't really work well for gliomas. It worked very well for CNS lymphoma. There are some other approaches being used now using focused ultrasound or LIT. It's still a little unclear how much access you really get with those approaches. That is work that needs to be done in more detail. And then there's this area of direct brain delivery. And the concept of direct brain delivery, putting the drug directly in some sort of release mechanism, was actually validated by Gliadel, the carmustine wafers. Of course, that predated the development of temozolomide, and the effect was modest, and they have overlapping activities, so temozolomide has become the standard. The idea of doing intrathecal or intraventricular injection has been problematic because the CSF is not really in homeostasis with the interstitial space in a way that gives kinetics that are likely reasonable for the therapeutics. And then there's this idea of convection enhanced delivery. CED was developed initially by Ed Oldfield at NINDS. The idea was that you would pump a drug of interest slowly into the brain. Now, that seems kind of obvious today and the like, but back then, and when this was conceived of, most of the neurosurgery was worried about how to get fluid out of the brain because that was a bad thing. And so the idea of actually pumping fluid into the brain was completely counterintuitive, but it actually worked because if you go slowly, you don't actually increase the intracranial pressure, and you actually get better distribution, more uniform distribution than just with diffusion. And so this schematic kind of illustrates the options that we have for introducing drugs directly in the brain. One is to do an injection. And when you do that, the brain is viscoelastic. It kind of resists injection. Most of it comes up around the needle. We don't really show it here. Most of it goes up this way. This, of course, is a resection cavity. These are the tumor cells that are in the infiltrative margin. You're not going to get much coverage that way. And there have been some recent clinical trials that have used that approach with replicating viruses and haven't really worked out. There was the wafer approach, which I mentioned earlier, which did provide proof of principle evidence. But again, it's a diffusion-based mechanism. You're not going to get much penetration of the drug of interest. And then the idea is convection enhanced delivery. Now, it's easy for me to make a schematic and show this and say, hey, see, it works. You can do that with anything. I'll show you data later on that I think are going to be more convincing. So what can be delivered? Almost anything, conventional chemotherapeutics, but they must not cross the blood-brain barrier easily, which is good because there aren't many that do. The problem is actually now the reverse. You want to keep them in the brain. So you want things that don't cross easily. And most conventional drugs don't cross easily. The small molecule drugs could be, but on the other hand, if there are substrates for the active pumps that are part of the blood-brain barrier, they'll just get pumped right back out. I like the idea of nanoparticle formulations. I actually saw a patient earlier today who was asking me all about that. I think the idea of trying to introduce drug depots in the brain would be a great idea. It's just there are challenges in the formulation and the approval process. Oncolytic viruses, vectors, all sorts of things can be delivered this way. There have been a number of clinical trials that have looked at using CED, and these are the different classes of drugs. Many of them were created specifically for this approach. Some of them are old chemotherapies that have been used, and you can see the different phases of trials. There's only one that made it to completed phase 3. That's this one here, the Centritic and Vasutotox. This one was actually aborted, but this one actually made it. And it ended up being a negative trial. And there were a number of factors that led to this. I was part of a failure analysis that really tried to look at this question. And there were a couple of important principles that we decided were very important, obviously, which was that we really couldn't do pharmacokinetics. We were just pumping a drug in with a tube that we pulled off the shelf, and we had no idea where it was really going. At that time, we could not co-infuse a tracer easily. And we had questions about the technology, since it wasn't really, we knew that backflow was an issue. And here's that example I wanted to show where you can see when CED works, it really works. So this was, so John Sampson was able to co-infuse radiolabeled albumin in that trial with some of the patients. And here you can see a catheter, and this is after 48 hours infusion. Beautiful infusion here, nice distribution. Here's the resection cavity. And so you can see there are a couple of problems. Number one is this area is not getting treated at all. This area is not getting treated at all. This area is not getting treated at all from that one catheter. But it showed that, this can work. Unfortunately, most catheters look more like this, where you had some initial infusion, but then the rest backflowed and was just over the surface of the brain and got nowhere. And the problem was we were just using tubes off the shelf, and we knew that those tubes were not really up to the task, but we didn't have enough evidence to prove that. Once we generated this, it became less interesting to use a tube off the shelf. And that was an opportunity to try to develop better technology. But the other thing was we had to be able to see where the drug was going. There were a number of technical and regulatory hurdles that made that nearly impossible. As is usually the case with progress, it was a patient with a connection to a U.S. senator that ended up breaking that logjam, and the FDA suddenly was allowing a tracer to be co-infused. There had been lots of preclinical work showing it was safe. So it ended up, Russ Lonser was able to do that and show that you could safely use it. Also, as I said, we knew that the off-the-shelf catheters were not really up to the task. This is work that, again, had been done early in the development of CED, showing that the size of the tube really mattered, actually the end port. And that was where the idea of a step-down catheter came from, having something very small. It does have its own issues, plugging during insertion and then introduction of air during stylet removal for a non-rigid catheter. Those are all problems, but at least the principle was there that we needed to go small. And so there were a couple of new devices that were developed based upon that principle. The step-down was first developed by MRI Interventions called Smart Flow Cannula. There's a second version of that that's more flexible, that's made by Brainlab. There's a MEMS cannula made by Alcyon, a chronic one made by Renishaw, and then I'm going to talk more about the one that we developed when I was in Cleveland. Some of these have 510K or CE mark clearances, none for its use as CED devices. We don't need to go into details of why that is. Also, it's important to know that this one here, the SmartFlow and the MEMS cannula, are rigid devices, can only be used in the OR, which means very small volumes of infusion, whereas the Brainlab and the Cleveland multiport can be used over a period of days. This one has only been used in the UK to date and is really a custom-made system for each case. It's not yet available commercially in the US. So I've actually had the opportunity to use the 4 that are commercially available. This is a case that I did in intraoperative MRI with the MRI Interventions device. This was during the Tokagen trial. And we actually did CED of that virus. This is actually work from Mark Suedane, who's been developing a theragnostic for treating DIPG, and he's been using the Brainlab version of the MRI Interventions cannula, and really exciting work that he's been doing there. But ultimately, when we start thinking about glioblastoma, we have to think about the volume issue. Okay, so when we're treating, we're not just treating a rim here. We're not just treating this enhancing tumor. We're treating this whole area. This is where we need to get drug to. And so if you think about that, you have to think about what is that T2 flare volume. That's just a start. It goes beyond that. What is that volume? So that median volume, we looked at this in a series of patients that we were evaluating volumetrically with GBM. it's about 37 cc's median, but it's as high as 170 cc's. So you need to really be able to cover a very large amount of brain. And when you have a single source device that can be used in the OR only, and an infusion rate at really the maximum is about 10 microliters per minute, the most you're going to get over 6 hours is 3.5 cc's. And so if you think of maybe a 2-to-1 ratio for that, maybe 3-to-1, you're not talking about these kinds of volumes. So we need something that we can, where we have time for delivery. So we set out to develop this device to achieve a reliable high volume drug delivery, not just for intraoperative use, that's backflow resistant, biocompatible, drug compatible, and can be used with conventional neurosurgical equipment, not just intraoperative MRI. This is a device we came up with. This was when I was at the Cleveland Clinic. We partnered with a global manufacturer, Parker Hannifin, and we formed this spin-out company called Infusion to develop it. It has multiple patents and most importantly, a 510(k) clearance. But making a device was not enough. I wanted to prove that it actually works and really understand how best to use it. So we put together this pilot trial strategy. We had to pick a drug. After all, there is no drug that's approved for direct delivery into brain tissue. And so we picked topotecan. This was based on Jeff Bruce's phase 1 work that he had done. It made it much easier. The CDER, the FDA liked that. We used gadolinium as our tracer, our device. We actually had some funding to do this. Managed to deal with all the conflict of interest issues. The biggest challenge was to get the device people and the drug people at FDA to talk to each other. They wouldn't talk to each other. We actually had to force the issue. They finally did. And we got the trial up and going. And this first pilot study we published recently in Journal of Neurosurgery, just where we put one catheter into enhancing tumor, one into tumor-infiltrated brain, same infusion rates, and we saw complete differences in extent of volume of distribution as we expected. We then went on and did a second protocol where we took out that enhancing tumor, since we know that's an effective treatment, and then put 2 of the catheters into the tumor-infiltrated brain, and now started getting volumes of distribution that were unprecedented. And that was a lot of fun to be able to do that. Now what we're doing is we're going back to the enhancing tumor, and we're infusing at a much higher rate, and we find that we can overcome the leakiness of the blood-brain barrier and actually get distribution not only in the enhancing tumor, but also the tumor-infiltrated brain. This is one catheter that was used in this case. We've done 2 of them now, and we're looking to go beyond that. All right, so now let's talk about therapeutic development. This is really the exciting part. So now we have a device we know that works, and really we need to start thinking about maybe better drugs. Topotecan, I think, has some positives. It also has some potential negatives. Let's try to really target the biology. And so we're working together with a company called OncoSynergy. It's a biotech startup that's led by Anne-Marie Carbonell. Anne-Marie was actually a neurosurgery resident with us in Cleveland, who ended up via a few steps becoming a leader in biotech development. And so she and I put this trial together. When I say she and I mean she. Really, took their lead compound, which is OS2966. It's an antibody that targets the master integrin subunit and involves a number of interesting design features to the trial that we both were excited about. One is direct delivery. Second is the window of opportunity design to really look at what's going on. And so this is actually a slide that she provided. Where we're initially doing this first-in-human study with the usual sort of objectives that you would have with a first-in-human study, but also with the pharmacodynamics. These are recurrent GBM patients, the usual criteria. We're going to start by doing a biopsy, and so we'll have pre-treated tissue. We'll then do an infusion into the enhancing tumor, cover that, as we showed we can do now. And then we'll come back a day or 2 later and resect the tumor. And now we'll have a treated sample to look at. And then we'll go ahead and treat the tumor-infiltrated brain as well and have typical clinical endpoints. Again, this is a timeline showing that in more detail. And this is just some examples from our initial studies when we were validating the device. We've just opened this trial, the OncoSynergy one, and hoping that we'll have our first accrual shortly. And as part of that analysis, Anne-Marie and her team have a detailed understanding of what kind of markers to look at when they use this drug. They've done a lot of preclinical data and evaluations of this. And so they'll be able to look at the ability of the drug to bind to its receptor, and then look at downstream signaling, and then, of course, ultimately functional assays as well. And then we'll have the usual clinical outputs.